So let's go ahead and talk about how muscle actually functions. And when we're talking about the function of the muscle, we're talking about how do we get the muscle to contract. This is what we have to remember is that the contractility of the skeletal muscle is all about its voluntary recruitment. And it's going to involve several components within the cell itself, which is going to lead to the shortening of the sarcomeres, which is seen by the I and H bands disappearing as the Z lines move towards each other. That's the evidence we have to muscle contraction by the sliding filament theory. However, we have a secondary component to this in which we know that the entire muscle fascicle and the entire muscle fiber and the entire muscle itself, the length of that whole unit is going to contract. It's not just the individual sarcomere. The way in which this muscle contraction takes place is based off of the combination of the excitation contraction couple, that's the depolarization of the membrane, and the sliding filaments within the cylindrical myofibril within the cell itself. The way in which we talk about this contraction is based off of how the fiber itself is contracting. That is, what is the rate at which we get depolarization taking place, in which we talk about it based off of being what's referred to as a twitch. That is a single contraction of a single muscle fiber. Trep contraction or fused twitch. This is where we have repeated twitches within the fiber based off of a summation of recruitment signals. And then tetany or tectonic contraction. That is where the entire muscle fascicle is in a sustained contraction. We do not have any separations of twitches. Regardless of how we discuss this, it's all going to start at the neuromuscular junction. The neuromuscular junction is the interaction point between the motor neuron and the muscle fiber itself. In the muscle fiber, we have invaginations of the membrane, the sarcolemma, at what's referred to as the motor end plate. This area has a high density of acetylcholine receptors and a huge amount of sodium channels. Near the acetylcholine receptors, these are going to be ligand-gated receptors. As we move away from the motor end plate, they become voltage-gated channels, which means that we have to reach a threshold in the membrane potential in order to get those sodium channels away from the motor end plate to open up. And so if we look at the motor end plate, this is what we're looking at. So we're looking at the end of the motor neuron coming in, the synaptic knobs with their acetylcholine vesicles. When depolarization passes through that motor axon, it's going to allow for release of acetylcholine. That acetylcholine will then bind onto the receptors on the sarcolemma at the motor end plate, allowing for depolarization in that region. And the depolarization will then spread via voltage-gated channels. Within this, we have two distinct functions that are going to take place. We have a time issue in between when we get stimulus and when we get action potential. That's the latency period. And then we have the time in between when I've hit my potential and gone through my action potential and when I can go about getting repolarized again. That is, can I get stimulated again? The refractory period. In this, we have a very, very short latency and a very, very short refractory period in skeletal muscle. It's so short that the timing between when the action potential hits and when I see contraction, around 2 milliseconds, I have already started and entered into my refractory, which means that I can go about getting a secondary contraction taking place within the contraction cycle that has already started. Because of this overlap in between latency and refractory, we can sum the individual twitches within the muscle of the skeletal muscle tissue, leading to sustained contractions. The way in which we're able to get to this sustained contraction, or even this twitch contraction, is based off of the all or none principle, which means that we're not going to have any partial response. We may have a graded response within the muscle whole. However, the muscle fiber itself does not have a graded response. It's all or none. Either I reach threshold or I don't. If I reach threshold, I get action. If I get action, I get contraction. The amount of stimulus that is necessary for the fiber to get to threshold will vary based off of the diameter of the fiber that is there. Where we have low threshold fibers, which are going to be small, and high threshold fibers, which will be large. Each contraction that we see within this response is referred to as the twitch. We're able to develop variable levels of mechanical force or strength based off of, one, the diameter of the fiber being recruited, and two, the number of fibers that are working at a given time. The way in which this recruitment takes place